Pooja's here. It's really nice to be here and see a lot of our phage crowd here at UC San Diego. As some of you might know, I'm Katrina Weitzen. I'm coming up from UCI, and David Pride and I have been organizing the IPATH seminar series this year, which has been wonderful. And today I have the pleasure of introducing Professor Breck Duerkop, who's joining us from Colorado. And he has a really interesting background that prepared him really well for studying phages, which included a bachelor's degree from the University of Wisconsin in Milwaukee. And then I saw that you worked in a DNA sequencing facility around the year 2000, like
Yes.
right when the human genome was sequenced. That must have been a really cool time to work in a sequencing center.
And we were still doing slab gels.
Wow.
Automated slab
All
gels.
right, so there's probably almost no sequencing technique you haven't tried in that case. That's cool. And then Breck moved to the University of Washington in Seattle, where he did his PhD with Pete Greenberg, who is somebody who I've read a lot of their work because of the Pseudomonas and cystic fibrosis connection. So that must have been a really nice place to do a PhD. And then Breck moved to Laura Hooper's lab in Texas, where he had NIH funding both for E. faecalis work in F32 and also for phage work at KO1. So that seems like a really cool combination. And his research program is still very much focused on the Enterococcus faecalis and phages and the interaction with the host. And he has NIH funding around a lot of those ideas and even some more practical ones. I saw you had more business-focused grants too. So it seems like really fun. And Breck is regularly publishing in society journals. His papers are showing up on the medRxiv and the bioRxiv first, which is really cool. And one of our most recent visitors, Daria Van Tine, I noticed you guys have a really interesting preprint about E. faecalis phages up just from the last few months.
Yes.
Okay, so I will not belabor it. I'm really excited to hear about what you've been up to lately, and thanks so much for coming.
Okay. Okay, great. Thank you, Katrina, for that really wonderful introduction. And so it's really my pleasure to be here today to talk with you guys about everything surrounding enterococcal phages and what's been going on in my lab for the last 6 to 7 years since its inception at the University of Colorado School of Medicine, which is located near Denver in Aurora, Colorado. So my lab really focuses and centers our work around mobile genetic elements with a major focus on gram-positive enterococci. And we study elements like transposons, plasmids, but also phages. And really phages sit at the center of what I'm going to tell you today, but ties together a number of these different types of ideas that are related to how phages interact with mobile elements in these bacteria of medical concern. And so to give you a little bit of a primer as to who the enterococci are, so these are antibiotic-resistant bacteria of medical concern. They're gram-positive commensals. They're primarily minority members of the gastrointestinal microbiota, where they pretty much live in harmony with most healthy individuals. However, they become problematic when people are immunosuppressed or undergo really harsh, broad-spectrum antibiotic therapies, where the enterococci can bloom and become a really abundant component of the microbiota and take over this site of natural colonization. And this can then lead to a number of complicating factors in individuals that are predisposed to having such complications, such as bloodstream infections, urinary tract infections, and even in some very extreme cases, the enterococci can cause really serious threats to the health of your heart in the form of endocarditis. And so the enterococci, being bugs of concern, obviously fall within the escape pathogen classification, and they're a real common cause of nosocomial infections, primarily caused by E. faecalis and E. faecium. These are acquired in the hospital through contact-dependent exposure or from patient to hospital staff transmission. And they're resistant to a large repertoire of different types of drugs. specifically last-line drugs, such as vancomycin, and even some of the more contemporary drugs that we use today to treat really difficult enterococcal infections, such as daptomycin and linezolid as an example. So just to give you some perspective, at the University of Colorado, about 70 to 80% of our enterococcus healthcare-associated infections are antibiotic resistant, and many of these are MDR, specifically vancomycin resistant and ampicillin resistant. So these are really problematic bugs that are hard to treat. And this comes from the fact that they are capable of horizontally transferring their genomic DNA, horizontally transferring mobile elements amongst each other, which can provide certain degrees of resistance through the mobilization of multi-drug resistant plasmids, horizontally acquired elements such as prophages, so on and so forth. And because of this plasticity in their genomes, this is really what makes them very adaptable to thriving in the hospital setting under various selective pressures. So for this crowd, I don't need to belabor the point that, you know, we're really failing in developing new drugs to target multi-drug resistant bacteria. And I would just like to point out that, you know, we really are sitting in a discovery void when it comes to new drugs to target these difficult to treat organisms. And there's really not a lot of push by the pharmaceutical industry to really make this change, right? And so the takeaway is that antibiotic development is really left to smaller research groups, which just is not fiscally possible for many research-intensive groups. And so that's really what brings us all here today, and the spirit of IPATH, right, is the fact that phage therapy is gaining traction as a real viable option that could be utilized as a monotherapy or potentially as a therapy in combination with antibiotics as a way to treat these difficult to resolve bacterial infections that are drug resistant. And so I won't spend a lot of time telling you why these viruses are so great, because you all know why. But what I will tell you is some of the ways that we think about phage-host interactions in the context of developing new knowledge that could be helpful for the development of phage therapies in the future. And what we really focus on are some of the overlooked aspects of phage-host interactions as they relate to how bacteria respond to phage infection at the genetic level, and how phage-host interactions influence the evolution and integrity of bacterial chromosomes. And so in my lab, the overarching goal is to really understand how enterococcal fitness and pathogenesis is influenced by phage infection, which will help hopefully inform future phage therapies to come. Okay, so what I'm going to tell you is 3 short vignettes of work that's been going on in my lab. The first centers around a screen that Sydney Johnson, a former PhD student, and Anushla Chatterjee, a former postdoc in my lab, did, where they really just asked a simple question. We'd isolated a number of phages that came from wastewater that could infect E. faecalis strains that we have in our collection in the lab, and we just asked, well, what's the diversity of how specific are these phages for particular strains, and is there some that have broad host range and some that have narrow host range. And so what you can see, I think, and appreciate here is that these phages shown up here at the top have infectivity for a number of different strains within our collection. Some phages, like phage 4 or phage 43, for instance, have broad host range, whereas others, like phage 47, to give you an example, have a very narrow host range. And this is really just a presence-absence plot showing you if it's pink, they're highly virulent against that particular bacterial strain. If they're light pink, they're more weak infection, and blue is no infection. So what we wanted to know is, how do these phages infect E. faecalis? Is the mechanism conserved, or do these phages have really diverse mechanisms of infection? So to cut to the chase, and to make a long story short, what we learned is that by taking a handful of phages from that collection on different Ephicalis strain backgrounds, we generated spontaneous mutants that arose after growing in the presence of phage. And what we found was that in all cases, mutations that arose that conferred phage resistance fell within a large cluster of genes that encode the EPA exopolysaccharide, known as the enterococcal polysaccharide antigen, that's on the surface of the Ephicalis cells. And importantly, the mutations all fell within these genes at the bottom here that are colored in black with the stars above them, which compose of the teichoic acid specific decorations that are added to the ramen backbone of the exopolysaccharide that's embedded in the cell wall. And so these mutations are all sufficient to confer resistance to phage infection. And so we were interested to know, were there fitness trade-offs that happen due to acquiring mutations in EPA, and does that have a phenotypic influence on Ephicalis in the context of, say, its natural setting, like the intestinal tract? So what we did is we did an experiment where we put mice on water that contained Ephicalis in the drinking water, and in this case we used a glycosyltransferase mutant in this EPA-S gene, which is part of that variable region responsible for teichoic acid biosynthesis. And we colonized mice with these antibiotics in the absence of any perturbations. This is what we call our chronic or natural colonization model, where we don't have to disturb the native microbiota. We can introduce the bacteria to the animals through at-will feeding within their drinking water. And then over time, we'll get stable engraftment of the bacteria in the gut, and then we can wash out we do a washout period with regular water, and then we can assess colonization levels and manipulate the bugs by, say, adding antibiotics and so on and so forth. So what you see here is just colonization levels comparing wild type to this EPA-S mutant. And so clearly there's about a 2 to 3 log reduction in colonization efficiency of the EPA-S mutant compared to wild type, indicating that EPA is important for more stable colonization within the intestine. But more importantly, in my opinion, when we then challenge these animals with vancomycin, a single dose of vancomycin at day 21 here, you can see that the wild type population blooms to very high density, as you might expect, whereas the EPA mutants actually fail to bloom, and they only begin to outgrow, we believe, after some of the natural microbiota within that population has been knocked down from the vancomycin therapy. So we have done, in data that I'm not going to show, we've shown that these EPA mutants are more sensitive to vancomycin treatment by doing traditional MICs. And so this is a mutation that comes with a fitness trade-off, right? These bugs have acquired a mutation that allows them to overcome phage infection, but comes at the cost of their inability to colonize the intestine as well and outgrow in the presence of antibiotic selection. So we think of this as a potential means where we could potentially use phages as a tool to start steering these communities towards phenotypes that make them more medically tractable in terms of applying current and contemporary therapeutics. Okay, and we also tested whether or not does this just matter for colonization, or can we actually influence the ability of these bugs to be transmitted from one host to another? So the only way that we could really think of setting up an experiment like this was to do a similar type of feeding experiment, but this time using pregnant mothers. These pregnant mothers are exposed to the bacteria containing drinking water during the entire gestation period, and then right before they give birth to their pups, we do this washout period with regular water, and then we assess colonization levels within the pups that are born to these mothers. These mothers have GI-associated enterococcus, but the pups after birth are never exposed to the contaminated drinking And you can clearly see that at 1, 4, and 7 days post-weaning, you can see that the wild-type bacteria are transmitted really well to these pups, whereas the mutant EPA-S deletion strain fails to colonize these pups after birth. And so not only is EPA important for colonization and growth within the intestine, but it's also important for host-to-host transmission, which we think is a pretty cool observation, because one way that people acquire, VRE in the hospital is just through contact-dependent transmission. Okay, so what's the mechanism? How is EPA resistance conferred within these cells in the context of the broader, question of the genetic mechanism? And so in a number of situations, we get single nucleotide polymorphisms that have, deleterious effects on probably the enzymatic function of these exopolysaccharides specific, like glycosyltransferases, for instance. But in a certain population of resistant mutants, we actually get insertion elements that basically hop into EPA-associated genes. And in this situation, what I'm showing you here is an experiment where Josh Kirsch, a PhD student in the lab, isolated a number of spontaneous mutants for this individual phage that we call phage 19 that were capable of surviving during phage infection. And what he saw was that this particular IS element, called IS-256, was inserting at a very specific location in the EPA-X exopolysaccharide glycosyltransferase that's part of that variable region that I showed you a few slides before. And this element inserts in both orientations on the top and bottom strand of the chromosome, and it can be measured using just standard PCR specific for a portion of the EPA gene and the IS element itself. And we do not see these types of insertions at this location as you might expect in wild-type cells. So you might ask yourself, well, what is an insertion sequence? Well, it's really kind of the most simplest version of a transposon that exists. It's a transposase gene that is actually flanked by 2 inverted repeats. And these are medically relevant elements due to the fact that they often flank DNA, largely antibiotic resistance cassettes, and create what are known as composite transposons, which basically carry cargo. And the cargo in between 2 inserted IS elements can actually be excised, mobilized, and moved from one region of the chromosome to another as a means of diversifying genomic content. So you might ask yourself, well, why does this matter in the context of phage infection? Well, we were interested in this primarily due to the fact that a large number of clinical isolates of Ephecalis that have emerged recently based on draft genome sequences that we were able to pull from RefSeq have IS-256 associated with their genomes. Some of these are MLST types that are of significant concern within a hospital setting, which are shown here in red. And they're also associated with genes in these Ephecalis genomes that are associated with virulence, right? So it seems that IS-256 is a signature element that may define one particular genomic feature of multi-drug resistant problematic strains of Ephecalis. And so in order to get a better idea of how IS element mobility contributes to the genome biology of Ephecalis, Josh wasn't really satisfied with kind of our single isolate top-down approach of assessing IS acquisition in single colonies. And so what he did is he generated Illumina sequencing libraries where he basically took advantage of a really simple technique of modifying traditional transposon sequencing to follow and track IS element mobility in real time in these bacteria. So the way this works is he makes a next generation Illumina library using his favorite Illumina library preparation kit. And then he does a PCR step, because in these situations where IS-256, shown here in green, inserts into a location within the chromosome, you're going to have the IS-256 terminus associated with some piece of DNA that's specific to the chromosome. And after adding Illumina adapters, you can actually use PCR to amplify off the Illumina adapter and the terminal end of the IS-256 element and actually get the junction between IS-256 and the host chromosomal location where it's inserted. And then you can just quantify this by read mapping. And so what I'm showing you here in black are the 6 canonical sites on the Ephecalis V5A3 genome where IS-256 resides based on the sequence genome from 2003 that was deposited by Tiger as a high-density resident IS-256 insertion. And I think what you can appreciate here in these 3 different replicates, that IS movement is pretty broadly distributed across the bacterial chromosome, to some extent. And when we started looking back at some of our phage-resistant mutants, what we found was that these 19RS strains that are resistant to phage 19 actually have, based off of whole genome sequencing, a higher number of predicted IS-256 insertions in their chromosome compared to the canonical wild-type V5A3 isogenic background. So what Josh did is he did IS-seq on some of these to confirm that there was higher levels of mobilization of IS-256 following phage infection. And as you can see here, compared to wild-type, there's a number of different peaks that emerge within these read mapping plots that indicate that there's not only new sites of IS-256 insertion, but also sites of IS-256 insertion that are of a higher density at locations where they naturally reside in the wild-type strain background. And if you do pairwise comparisons between these strains at every single peak across the chromosome, you can basically see that you have a higher abundance of IS-256 insertions and depth of density between phage-resistant Ephecalis strains compared to the wild-type V5A3. So Josh really wanted to know, is this driven by phage infection? And so he did an experiment that I thought was quite clever. He took a strain of Ephecalis called OG1RF. It is a kind of prototypical lab Ephecalis strain that many people have used, to basically make their careers in the field. And it's a really good model because it actually has very little mobile DNA in its genome. It lacks plasmids, it has no prophage, and it actually has no IS-256 elements. And so what he did is Josh cloned IS-256 onto a plasmid, moved this into Ephecalis OG1RF, and then infected these cells with phage and asked, do I get better survival? And you can see that we get better survival if there's an intact wild-type copy of IS-256 being expressed off of a plasmid. A catalytically dead transposase looks very similar to empty vector, so this delta DDE is catalytically dead, and you can see that survival rates are similar to what you see for empty vector. So this tells us that IS-256 is protective against phage infection. And he took it a little bit further and then made a translational fusion of IS-256 to GFP. And in the absence of phage infection, he compared GFP expression to during phage infection. And you can see there's an increase in GFP expression over time following phage challenge. And then a nice feature of these IS elements is the way that they move is they are a copy and paste mechanism where they basically retain the original insertion site and copy out a new copy of the IS element during transposition so that that piece moves to a new location and the original site is retained. And when they do this, they actually make a circular intermediate where the insertion element, as it's copied and excised, is basically circularized. And then you can do PCR at the junctions of these predicted circles to actually get a frequency of circle formation in these cells. Sorry about that. And you can clearly see that IS-256 circle formation on a per cell basis is enhanced during phage infection compared to no infection. So what this tells us is that phage infection drives the activation and mobility of IS elements to diversify Epicalis genome content. So how is this being done? So it turns out that these phage-resistant, or these 19RS strains, as we call them, are actually chronically infected with this phage, phage 19. So you can see when we grow cultures of these bacteria and we take supernatants, we can actually recover a large number of viable virus from the supernatants of these cultures from both log and stationary phase. We can PCR for specific genes that confirm that the phage is present in the samples compared to wild type, right? And if we actually take colonies from plates of these strains and we patch them on lawns of an indicator bacteria that would be susceptible to phage 19, you can clearly see that they're shedding virus from the colonies and forming these zones of clearing or rings around the colonies compared to wild type. And so it's about 10 to 20% of the colonies that come out of these cultures are actually positive for the shedding of these viral particles. And we believe that this is an important feature that the bugs use to compete in an environment where they have a susceptible competitor. And so you can see in this situation, if you take Epicalis OG1RF, which is sensitive to the virus, you co-culture it with any one of these 19RS strains, you can see that the 19RS strains have a very good competitive advantage in terms of their competitive fitness. However, if you make a strain of Epicalis that's resistant to phage infection by knocking out the glycosyltransferase EPA-LX, you can see that carriage of the phage now becomes a fitness detriment to these organisms. So there's a trade-off, again, in terms of maintaining these viral genomes within the population. In one situation, it can be used as a tool for competition, but it comes with genetic cost when these elements are not necessary for competition. Okay. So in conclusion to this first part, really what I've told you is that in Epicalis during phage infection, you get mutations that modify EPA. What I didn't show you is that this actually, the mechanism of resistance is really failure to adsorb to the surface of the cell and eject DNA into the cell. But cells that can successfully be infected by some phages, in particular phage 19, can harbor these phage genomes. We're not necessarily sure if we would really call these pseudolysogens or what they would really be called, but they can actually drive genetic changes within the bacterium through the mobilization of IS-256, which basically diversifies the genome. In one situation, these insertions land in EPA, which confers resistance to phage infection, but in a lot of other situations, these IS elements are moving to a lot of different places. And the genetic consequences of that are really unknown at this point. Okay. And my lab is also really interested in studying phage-host interactions in the context of ephesium as well. So as many of you know, ephesium is also a very problematic multi-drug resistant member of the enterococci. And we've been working for a couple of years now with Howard Hong, just down the street at Scripps Research, exploring some mechanistic biology behind how phage interact with the Epicalis cell surface. And so Greg Canfield, who was an ID fellow in my lab, set to isolate some novel phages that infected ephesium, and he settled on these three phages for the vast majority of his work. And one phage in particular, this phage that we call 9181, actually conferred mutations that all localized in an active site of a peptidoglycan hydrolase called SAGA. And this is really what connected us with Howard to really begin exploring how cell wall modification influences phage-host interactions in ephesium. And this is because in Howard's lab, they'd published a few years before that SAGA from ephesium is an endopeptidase that cleaves lyse-type peptidoglycan, releasing ramal dipeptide, which is a potent stimulant of intracellular NOD2 signaling in intestinal epithelial cells. And this signaling through NOD2 is really important for barrier protection at the mucosa during pathogenic invasion of non-resident bacteria. And so reading some of Howard's work and realizing that we are getting mutations in the same protein that's important for cleavage of cross-linked peptidoglycan, we made a handful of constructs and sent them to Howard's lab. And what we learned is that these mutant versions of SAG-A that we call 81R6 and 81R8 are unable to cleave cross-linked peptidoglycan at these positions indicated here by these scissors, right, to generate individual products shown here as product 1 and product 2, product 2 being, you know, your quintessential mirandyl dipeptide. And so you can see, or sorry, mirandyl dipeptide is shown here. So you can see in wild type, we see, you know, a high abundance of product 1, we see some product 2, but in these mutants, they look very similar to buffer, as you can see here. And so I know I realize this is a little hard to see with all these peaks, but you can see the loss of peak formation in these mutant strains, indicating that these peptidoglycan-specific hydrolases are not cleaving their target products properly. So a postdoc in my lab noticed that these cells had a very sufficient growth defect. So she grew these in standard laboratory media and showed that if she added SAG-A back on a plasmid, she could restore growth similar to what we see for wild-type Ephysium. And then she did a staining experiment, and I hope you can see this okay, where she used this dye called HADA, which is a blue fluorescent analog of D-alanine that gets incorporated into sites of newly synthesized peptidoglycan in the cell wall. And what you can see for wild type is that they form these triseptal staining patterns, as you would expect a diplococci to do. But in the 81R6 mutant, you can see that this is all around the periphery of the cell, and we can restore the triseptal staining by adding SAG-A back in on a plasmid. So she took EM images of these bacteria, and you can see that these diplococci in wild type have very, you know, clean morphology. They have nice, you know, division septa that are forming in cells as they're actively dividing, and these are just two examples of wild type cells. And you can see in the 81R6 mutant that you have very aberrant, you know, basically swelling of the bacterial cells. You can see that their division septa are very long and non-symmetrical. And you can even see that in some cases there's little mini cells that are kind of, you know, coming off of some of these dividing cells here. And so this shows us that, you know, SAG-A is fundamentally important for the proper cell, you know, morphology of diplococci Ephysium. And Ephysium SAG-A catalytic mutants actually have cell wall defects, and we tested this by simple dye uptake assays, where in one situation we take advantage of a derivative of beta-galactosidase that is chlorophenol red beta-galactopyranidase. And when this is cleaved by endogenous beta-galactosidase itself, you get a red color that forms. And you can see here that there's an accumulation of dye at an OD570 in the mutant strains, but not in the wild type strain. And then if we use a fluorescent form of penicillin, which should be able to penetrate these cells to some extent, and target PDPs and label them by fluorescence, you can see that the 81R6 SAG-A mutants take up a lot more bacillin on a concentration-dependent series here. So why do we care about this? Why do we care that these cells are really sick and that their cell walls are, you know, aberrant? Well, we've done MICs on some of these, and as you can clearly see in the case of, say, something like ceftriaxone, that we can actually reduce the MIC to pretty significant levels of antibiotic susceptibility here. And you can see that this is the case not only for ceftriaxone, but also for ampicillin as well. And so what we've really been interested in determining is whether or not we can then use these phages as a conventional antibiotics to clear bacterial populations. And so we've done some experiments where we're well below the MIC of wild-type Ephesium for things like ampicillin and ceftriaxone. And I think what you can appreciate here is that the combination of low levels of ceftriaxone along with phage infection is very effective at killing Ephesium compared to phage alone, shown in the open blue squares, or obviously drug alone. So the conclusions for this part are really that we're getting phage resensitization, we're getting resensitization to antibiotics through the emergence of mutations that confer phage resistance. And now the question really becomes, well, what's the mechanism? So we have some hints as to how this might be happening based on some of those EM images that I showed you. And our favorite hypothesis right now is that we're getting dislocalization of critical penicillin binding proteins, or potentially a combination of PBPs and components of the divisosome that are important for proper cell division, which render these cells more susceptible to drug. And so we'll be kind of tracking that down at more of the molecular and cellular level moving forward. Okay. So the last thing that I want to touch on extends to some work that goes beyond phage, the connection of phage and transposons and their connection, and focuses on plasmid biology and its intersection with the phage biology of the enterococci. And this is because plasmids are a really abundant source of mobile DNA within the enterococci in general. So the enterococci carry both broad and narrow host range plasmids that can be conjugated, and in a lot of cases they carry drug resistance genes that are important for their emergence as opportunistic pathogens. And this project really centers around the work that was started by Sydney Johnson in my lab prior to her leaving, and has now been picked up by Shelby Anderson, a new PhD student in the lab, in close collaboration with John Whitney's group and his PhD student Nathan Bullen at McMaster University. And it all comes down to this one particular mobilizable plasmid in E. faecalis called PTEF2. So this is a 57,000 base pair plasmid. It has a lot of the quintessential genes that are important for replication, conjugation, so on and so forth. But all these genes in black are hypothetical genes of unknown function. And Sydney made an observation early on when they were starting to do this work, that in this particular strain called SF28073, which is highly susceptible to phage 47 in our collection, V583 is resistant, yet their genomes are 95% identical. So the question is why? Why are they resistant? So the punchline here is the plasmids, right? So SF28073 has 3 unique plasmids that are genetically distinct from the 3 plasmids found in V583. And lucky for us, we've acquired a large collection of strains from people like Gary Dunny and Mike Gilmore over many years, who've been very generous to provide us with all sorts of cool strains that they've generated in their labs. And one of these strains turned out to be really useful and important for this project, and it's called V19. And V19 is actually a plasmid-cured derivative of V583. And you can see here that similar to SF28073, V19 is equally susceptible to phage 47 infection, whereas V583 is not. And so if Sydney transfers or mates in PTEF1 or PTEF2, which are 2 of these plasmids that are endogenous to V583, you can see that PTEF2 does not provide protection, PTEF1 does not provide protection, whereas PTEF2 does provide protection. And if she moves this into a different strain background, the strain called JH2-2, you can see that PTEF2 is also protected. So this is not just a strain-dependent phenomenon, but it really seems to be more broadly, or more focused on the biology of the plasmid itself. So lucky for us, Gary Dunny's lab had done a lot of work on an analogous plasmid that's very closely related to PTEF2 called PCF10. They're about 50 to 60% identical at the nucleotide level. And if you align them, you can clearly see that there's two major regions of variability between the plasmids. And it's this variable region, region 2, that's missing in PCF10 due to the insertion of this tetracycline containing transposon TN925, where we've centered a lot of our attention on a large cohort of hypothetical genes that fell within this region. But that's a lot of genes to tackle, right? I mean, we can do genetics in Enterococcus, but it's not trivial, right? So clean deletions was off the table. So lucky for us, Kim Klein's lab several years ago published a really beautiful paper developing tools to do CRISPR-I knockdowns in E. faecalis. So we acquired these tools from Kim's lab. And the way that this works is there's two plasmids in the system that are controlled by a nisin-inducible promoter. One plasmid carries a dead Cas9, so it's catalytically dead, but can still bind DNA. So it'll bind DNA, but it will not actually cleave, even though the guide RNA is present. The other plasmid uses the same promoter to drive expression of a specific guide RNA, and when used in combination, you can actually target transcription and knock down gene expression in a targeted manner. And so Sydney did a really straightforward screen. She generated this, she moved all these tools into V19 carrying the PTEF2 plasmid, and then she just plated cells on either agar that was selected for antibiotics and had nisin in it, or the same agar composition, but also carrying phage. And then she just looked for relative viability. So in the case of an organism that carries PTEF2 that is resistant to phage infection, you'd expect a relative viability around 1. And if you then can look at all the genes that she targets, we find two culprits here that limit viability when these genes have been, their expression has been knocked down. And these are in open reading frames 58 and 59 on PTEF2. So these are on the same transcript. So she just did some real simple RT-PCR off of cDNA, and we realized very quickly that this protein 58, or the transcript of 58, when targeted, is likely having polarity on 59, because if you actually make a clean deletion in the open reading frame 0059, you can restore phage infection, whereas the 0058 deletion has no effect on the ability of the phage to overcome infection. So our culprit here is this protein that I'll just refer to from here on out as 59. So what is this thing? So 59 is related to type 4 restriction enzymes. This protein itself, SSPE, has a specific domain of unknown function called DUF262. These are type 4 enzymes that are found in Vibrio species. And then this DUF1524 is a second DUF associated with type 4 restriction enzymes that's also found in the SSPE protein. And you can see these alpha fold structures of 0059 in blue overlap very nicely with both the SSPE DUF262 shown in red or the DUF1524 shown in orange. And so this indicated that we're dealing with a restriction enzyme that likely targets phage DNA directly. And so type 4 enzymes are enzymes that have evolved over time that are a little bit different than your canonical restriction system. So type 1 and type 2 systems, for instance, modify host chromosomal DNA and protect it from the enzymatic activity of the enzyme that would then target naked, unprotected phage DNA. In the case of type 4 systems, so then of course phage are real, they're resilient, they figure out ways to get around those restriction systems, and at the end of the day, they modify their genome so that they can't be cut by traditional enzymes. And bacteria use type 4 enzymes to target modified DNA. So we were interested to know if these enzymes were important for toxicity. So we spent a lot of time trying to try to set up experiments in E. faecalis using plasmid-based approaches to assess toxicity with and without phage infection. And to make a long story short, we moved to E. coli, which just gave us a little bit more of a more tractable system. But I think what's important here is that if we express 0059 in E. coli, at the point of induction, shown here by the arrow, you basically get a halt in growth. Whereas if you make catalytically dead mutants in both the DUF262 catalytic residue, this glycine shown here, or this histidine in DUF1524, you can see that these cells now grow similar to the empty vector control. And this is also more soluble protein is being produced in these cells. So we tag the protein with VSDG, and do Western blots, and you can clearly see that these catalytic mutants produce more of the protein compared to the wild type cells, which really does indicate to us that this expression of the protein in these cells is quite toxic. Okay, but like I said, these enzymes recognize modified DNA. So the question became, is phage 47 DNA modified? And typically the first thing one would do is just ask the question, is the simplest modification involved? Is it methylation, right? So we can identify methylated DNA in bacteria and phages and whatever your favorite DNA really is by doing bisulfite sequencing. And so the way that this works is if cytosines are not modified, the bisulfite conversion converts cytosines to uracils, and then when you sequence this by traditional Sanger sequencing, these cytosines come back as thymines. But if they're modified by anything, primarily methylation, but it can be other modifications at cytosines, these are then protected from conversion, and they sequence as cytosines as you would expect. So when we do this on E. faecalis chromosomal DNA, we get, as controls, we get conversion of cytosines into uracils, and they sequence as Ts. If we do this on a phage genome where we know we get really good protection of cytosines through beta-glucosylation, which is just a methylation modification where glucose has been added to a hydroxymethyl group on cytosine, you can see that you get full protection. And if we do it on phage 47 DNA, we get conversion to uracil. So this tells us that at least at the cytosines, in the traditional sense of the modification of a methylation at that site, that's not what we're dealing with here. So to make a long story short, we did a huge panel of restriction enzyme digestions on this DNA, and I won't bore you with all of the details, but basically we came up empty-handed. So at the end of the day, what we did is we turned to Oxford nanopore sequencing to see if we could get some inkling of an idea of what's going on with this DNA. So for those of you that aren't familiar with how Oxford nanopore sequencing works, basically you have a biological pore that's inserted into a charged membrane, and this actually then threads DNA through the pore, and since this is a charged membrane, as the DNA travels through the pore, a current can be measured as the DNA passes through the pore. And this is what's called the raw signal, or the squiggle. And these squiggles have been benchmarked against a number of different training sequences as this technology has been developed, and has basically been converted into a base calling strategy that allows you to decipher the sequence of the DNA that passes through based on these current profiles. And so if there's a modification in the DNA, those current profiles change, and then the base calling software will likely call those bases as an error. So in this case, it's showing this cytosine being base called incorrectly as a guanine. So what we did is we ran either Phi29 amplified phage 47 DNA through a nanopore, or the native phage DNA that was purified from, viral capsids. This 529 amplified DNA, in theory, shouldn't have a modification because that modification would be lost during amplification. And I think what you can appreciate here is that amplified DNA sequences really nicely based on these nice slanted curves, whereas you can see the native phage 47 DNA sequences very poorly. And not only are there more failed reads, there's actually more charge reversals where basically the molecule starts going into the pore and it gets stuck. And then when it gets stuck, the pore just basically pushes the molecule back out, and that's then recorded as an event that can be quantified. And so at first we thought, well, this must indicate that there's modifications all over the surface of this molecule, and this thing just can't get through the pore. But Shelby, being a very astute and savvy scientist that she is, thought, well, what if I just digest the DNA with a base cutter that can cut the DNA and then try it. And so you can basically see that when she digests with NHE1, now the DNA sequences more similar to what you see for the 549 amplified template DNA. So this told us that maybe our modification isn't necessarily a true base modification, but maybe it has something to do with the actual secondary structure of the DNA molecule itself. So then she went back and dug through some of the old, exonuclease, pages in the NEB catalog, right? And came up with these two phage-specific exonucleases, T5 and T7. And so T5 exonuclease is just what it is. It's an exonuclease that has double-stranded exonuclease activity, but also importantly, it has single-stranded endonuclease activity. So it can cut either single-stranded DNA or it can cut double-stranded DNA with closed ends. And T7 exonuclease is only specific for double-stranded DNA, shown here. So when she runs native phage 47 DNA on a gel, you get a large genomic band. She cuts it with NHE1, she gets the correct banding pattern. You can clearly see that T5 exonuclease can digest both species of DNA, whereas NHE1 cut phage 47 DNA is only susceptible to the T7 exonuclease. And so what this tells us is that the double-stranded DNA specificity of this exonuclease is not sufficient to cut this phage 47 DNA. And so what we think is going on here is we think these phage ends are closed in a single-stranded circle. This is just our hypothesis right now, and we need to do more biochemistry to test this. But this is akin to some really fundamental old genome biology that's been studied in E. coli phages many decades ago. And so what we think might be going on here is that these closed ends at the ends of the viral genome are what are complicating the nanopore sequencing, and the verdict's still out on whether this biology contributes to the enzymatic activity of our antiphage type 4 enzyme. But Shelby's working on that pretty vigorously, so I think she'll have an answer soon. Okay, so the last thing I'd like to finish with here is within the context of this restriction enzyme that's really potent at blocking phage infection. When Sydney was doing a lot of her experiments, she was always infecting with the same concentration of phage, around 10 to the 8 PFU per mil for consistency. And we started asking the question, well, our stocks are much higher than that. We have 10 to the 11, 10 to the 12 PFU. What if you just put a truck ton of virus on these PTEF2 containing B19 cells? Can you get escape phages? And so what you can see here in yellow is that we get a low proportion of plaques that can form on these B19 PTEF2 cells that basically can escape the restriction mechanism of PTEF2. And so she got about 9 plaques, and she picked these, and she sequenced the genomes, and basically they all had the same mutation. It was a G to A modification that in this gene called ORF65 that converted a leucine to a phenylalanine. And these AlphaFold images here show you that this phenylalanine, this bulky phenylalanine, sticks out the side of this predicted beta sheet here. What was really cool is if she ectopically expressed the L32F variant off of a plasmid in B19-PTEF2 containing cells and then infected with wild-type phage, she could basically restore infectivity by just adding this protein in on a plasmid in trans. And you can see the wild-type version has no influence, as you would expect. So we moved back to our E. coli system, and we co-expressed these proteins in the presence of 0059. And similarly, we could find that the L32F mutation, or this gain-of-function mutation, is sufficient to overcome the toxicity imposed on E. coli when expressing 0059, as shown in this salmon color here. And so what is this ORF65? Well, we don't really know. It has one domain that's similar to an SH315 domain containing protein. These are found in other enterococcal phages, and these domains are also found in ubiquitin E3 ligase-associated proteins that are involved in protein-protein interactions. So not a lot of information here, right? But what we did try is when we overexpress these in E. coli, you can clearly see that the L32F expression of this ORF65 L32F increases the amount of soluble 0059 that can be found in these cells, right? And more importantly, if we do a pull down, you can see that we pull down much more 0059 with the ORF65 L32F compared to the wild type. And so this suggests that this gain-of-function mutation in ORF65 is somehow contributing to the ability of this protein to probably bind to this restriction enzyme and render it inactive in some way. And so, more structural biology will need to be done to figure out where exactly these things are interacting and binding. But I will say we've tried to do, these kind of co-folding predictions and AlphaFold between the two proteins, and have just kind of fallen short on confidence with that data. But I think, we'll need to do more sophisticated analysis to figure that out. Okay, so in conclusion on this section, what I've told you is that these plasmids like PTEF2 contain, in this case, a phage-specific type 4 restriction enzyme that likely targets incoming phage DNA that's modified in some form, either through an actual base-specific modification or potentially due to some secondary structure differences in the ends of the phage DNA. And this ORF65 that is traditionally found in the phage genome has very poor specificity for 0059 in its native form, but when you have ORF65 L32F, gain-of-function mutation, that this can somehow bind and render this enzyme inactive. So with that, I really appreciate you guys taking the time to come and listen to the talk. I really need to thank my lab and the both current and former members of my group who have done the vast majority of this work. I need to really give a shout out to all of my wonderful collaborators throughout the United States who really make, the speed at which we can do this work possible and, make collaborative science just, a really fun activity on a day-to-day basis. And then, of course, my funding that supports the work that I presented today. So I'm happy to answer questions, and thank you very much.
Great. That was really a fun talk. Man, you have done so many great, using all the latest tools and the technologies we have. It was really fun to see.
Thank you.
Thank you. Okay, I guess
I have 3 questions about the first part.
Sure.
I'm very curious about. So when you found the resistant mutants, the first question is how long are they resistant? And is this related to when they get infected with the phage, is the level of the transposase higher or lower. So that's one, and the second one is like, are the phage particles that are being produced the same ones that you infect the bacteria with? Or I'm
Yes.
very curious about this.
Yeah, so I'll answer the first part of the question, which let me repeat it and make sure I have it correctly. Are the mutations fixed in the sense that do the phage that are the strains that are resistant ever become sensitive to phage infection? So to those phages, no. So these are fixed point mutations that are, genetically encoded within these strains and passed on to, daughter cells, right? So in that context, those SNPs are genetically fixed, and we don't see resensitization to those particular phages. Now, we've tried to screen for using those strains to screen for other phages that might be able to infect them, and we've had varied results with that. We do find some phages that can infect in the absence of EPA, or at least the modifications that are happening due to those simple glycosyl transferase mutations. But those phages have been really hard to propagate in the lab. So we can get plaques, but then when we try to do high titer workups on those in culture, it typically fails. So we can get low density composition of phages that can reinfect, but we got to do scrape plates from plaque assays and stuff like that. To your second question about the IS element, the IS element itself is more active, so we have some genetic evidence that supports that. So we have GFP reporter assay that indicates that fusing GFP to IS-256 during phage infection results in elevated GFP expression. We've also shown that these circular intermediates using quantitative PCR increase during phage infections. So those are two proxies that we use to assess greater IS mobility during phage infection. And now remind me your third question.
So you show that there are like phage particles being produced in the resistant mutants.
Yeah.
Are they like different prophages, or is it like the same one that you
Yeah,
challenge?
so I didn't make that very clear. So it's the phage that we use to infect initially. So it's this phage we call phage 19. And we have another very genetically similar phage in our collection to phage 19 called phage 4, and it does the same exact thing. So it's something unique about those phages particularly, and what we think is they're either lysogenizing, they're not integrating into the chromosome, so we've tested that. But they are maintained, and we don't know if they're maintained as an episome, are they maintained as like a lysogen, like kind of like a plasmid, right? Yeah, we just don't know. And we've actually, that paper's been through peer review, and one of the things that we got kind of reviewers got hung up on is they really wanted to know, like, is it really lysogeny? Is it pseudo-lysogeny? Or is it some kind of weird intermediate? And the simplest answer is that's hard to know, right? All I can say definitively is we have no indication that it's integrating into the chromosome like a traditional integrated prophage.
Hi, thank you so much. That was really an outstanding presentation. I'm not certain if I have a question. Maybe it's more of a statement and or clarification. But one of the things that we're really interested in here at IPAT is the idea that was really the main topic of part 2, I guess, of your presentation, and that is the cooperativity synergy between antibiotics and bacteriophages. And it was really nice to see what you have shown, and I had read that, but I think I appreciated it much differently when you actually were telling us about what you guys had shown. And one of the things, because we do see a lot of enterococcal infections, and one point that's really clear is that particularly a drug like ceftriaxone, nobody would ever think to use for an enterococcus, right?
Right.
And that's something that it will take a lot to get people to kind of choose to use ceftriaxone alone.
Right.
There is some data that indicates that if you use it in combination with ampicillin, that it can work. So that is going on, but it's really nice to see what you have, which is phage plus ceftriaxone working for Enterococcus faecium.
Yeah, exactly.
Yeah.
That's what, and maybe I didn't highlight that enough, right? Because that is what's really exciting to us, is that, the dogma really is like, you don't use cephalosporins because, low affinity PVPs are protective and you're intrinsically resistant, right? And in the case of wild type cells, yes, totally.
But so
yeah.
I mean, you've shown something that, can and maybe even should change some of the hearts and minds. I don't think it has broken through, but
Yeah.
sometimes things breaking through is just a matter of time. But that's really just an outstanding finding. Part of the question that I sort of have about it is, well, is that true for just one phage? Is that true for multiple different
Yes.
phages? And how reproducible is that?
Yeah.
Because it's just in general in clinical medicine, we kind of specialize in, we want to do this for everybody.
Right.
And, we don't like the concept of, test this and test that and test this. And that's one of the problems with phage therapy that we're
Right.
really interested in, is people don't want to test, but maybe
Yeah.
they have to.
Yeah, so at least 2 of those 3 phages that target faecium that I showed you synergize with ceftriaxone. The reason we focused on that one particular phage, 9181, is primarily because we're really fascinated by the SAG-A phenotype and some of the cell biology that we can do down the road with Howard's group to really get at mechanism of action. Like, how does mutation of that gene and obviously this very messed up cell wall result in antibiotic sensitization, right? And I think, by doing some genetics and some clever cell biology using fluorescence, localization of PVPs and other things, we can probably get to that answer. But yeah, there's other mutations that arise that are not in SAG-A, in faecium, that do allow ceftriaxone to synergize with the phage. Yeah.
I think the one last point of clarification I would try to make sure I understand it correctly is, we oftentimes think of ephesium, because ephesium loves Van A and Van B plasmids. But my assumption about what it is you show specifically with the phages plus the ceftriaxone, that should be independent of whether or not the organism has a Van A or
Yeah,
Van B.
absolutely.
Great.
Absolutely. Yep, definitely.
So there's a question from Zoom from Douglas Berg. He said, really intriguing. Do you know how phage 19 infection increases IS-256 transposase levels?
Yeah, so no, not really. We have some ideas. There's a phage-encoded anti-sigma factor that is in the genome, and we've done some ectopic expression of that protein in cells and then monitored IS-256 circular intermediates, and we see some increase, but it's been a little inconsistent. And the reason why we went after this anti-sigma factor is specifically in Staph aureus, this type of anti-sigma factor is fundamentally important for regulating the expression of IS-256. So IS-256 is not only found in Enterococcus, it's found in a lot of other gram-positive bacteria, but predominantly Enterococcus and Staphylococcus. And so that protein is a potential target, but I'm not fully convinced of the data yet.
Are there any other questions in the room?
Sorry, I think I just missed this, but why did you choose the NAT1 enzyme specifically?
Oh, I think there was no choice other than she used a sticky end base cutter. I mean, she could have used anything, I think, like EcoR1, whatever, right?
Okay.
Yeah, there was no reason other than that's what she chose.
So
Yeah.
just cutting the DNA relaxes those secondary
Yeah.
structures.
And it's probably because even though the secondary structure is on the end, if it's on the ends like that, but you cut in the middle, you at least have one site where now the exonuclease can get access to that, right?
Got it.
Yeah.
That's cool.
Yeah.
But you mentioned how this lab rat enterococcus that everybody works with doesn't have any of these mobile elements, and I wondered if that's something that happened through time. Do you know if it's something that happened through time because people were working
Passing
with
it.
it? Yeah.
So, it's funny because OG1RF, which is really like, most people in the field use, for wild type E. faecalis, if you're using a lab strain, you either use OG1RF or use V583. There's like two camps, right? And they come from totally different sites, right? So V583 is a bloodstream isolate from the 1980s, and it's VRE. And OG1RF is actually an oral isolate of E. faecalis that came from a tooth abscess. And if you think about the pangenomes of these strains, OG1RF and V583 couldn't be they're very different, right? Or I shouldn't say pangenome, is the wrong term. The accessory genomes, right? They have a lot of similarities in terms of core genome sequence, but they have a lot of differences in terms of other things that are, carried by them, and obviously a lot more mobile elements in V583. The idea is that in OG1RF, this is work from Kelly Palmer when she was a postdoc in Mike Gilmore's lab, is that certain commensal strains of E. faecalis that have natural CRISPR-Cas in their genomes are more protected from acquiring mobile DNA, right? So OG1RF has functional CRISPR-Cas. It lacks prophages, it lacks plasmids, and it doesn't really have many transposons, right? Whereas V583 doesn't have functional CRISPR-Cas, and it's littered with this, right? And this tends to be the trend that you see when comparing multi-drug resistant isolates to kind of your more bona fide commensal, right? And their work was really seminal in kind of proposing that evolutionary connection. And then I think it's been shown in other pathogens as well. I think they've shown it in Pseudomonas and maybe others. But I know for certain in Pseudomonas that seems to hold true as well.
That is very interesting.
Yeah.
Thank you very much. Is there another question?
I just wanted to ask you one last real quick question. Are you seeing any differences in trends in some of the things that you showed us today, specifically the synergistic interactions, when you look at a fecium compared to a faecalis, or are you just more focused on the fecium?
Yeah, we haven't done synergy with faecalis. We've done MICs on phage-resistant mutants, beta-lactam. So beta-lactams, as you know, are really good at killing E. faecalis, right? And so we don't see much difference in MICs, like a drop in MIC for phage-resistant mutants in faecalis against beta-lactams. But we do see it for daptomycin and for vancomycin. But we've never done the analogous experiments where we've used phage with, say, vanc or phage with dap, and then looked at synergy. And that's just for no other reason than we just weren't thinking about synergy at the time, and we kind of never went back and did those experiments.
Interesting. One last question.
Sure,
So
sure.
I loved your host range map and seeing how your different phages are doing. Were those myoviruses that have the broader host ranges, do you think?
Good question. 419 are myoviruses, and they do have a pretty broad host range. I'd have to look
419
back.
are the ones we did.
Yeah, exactly. No, 81. Yeah, sorry. There's too many of them, right? And they're all like these like silly numbers, which we need to get away from, I guess. That's a good question. I'd have to think about that. I have to look back at the data. I don't think we've ever really
dug into that too deeply. But the one thing I can tell you is in our collection, we pretty much only have cyphophages and myophages. We've never isolated a podophage, for instance, or any of these kind of more obscure, pleomorphic phages that are reported in the literature, but, yeah, we've never seen anything like that.
Yeah, I think we're kind of the same thing. We have this one podovirus, but it disappeared. Like when like a year or two later when we were working with it, we were doing EM. I mean, this is Puja's right here, and it happened in my lab too. Like around the same time, it was amazing that now it looks like a cypho. It used to be named and now we call it not
Yeah. But I know, like Bern, Schnabel, he's isolated a decent number of podos, I believe, right, on cytolysin containing the calyx, right? And I think published that in Nature, a couple years ago, right? And I think he's got some podophage in that collection, but then obviously some of your other normal culprits like cyphos and myos, right?
Yeah.
Yeah.
Yeah, interesting. Well, I think we owe a really big round of applause. Thanks very much for a great talk.
Yeah, thank you for having me. My pleasure.